Namaskar. Today we shall discuss about basic structure of chromosome for class 10 students. Here it's the second part of chromosome, that is what is actually the position of gene and where actually gene present inside the chromosome, or where is actually the chromosome present. If we will discuss about this, we will see that a cell, a basic structure is a cell. Often we know that our body unit is cell. A structure of our body unit is cell, and cell is what is the actually cell structure. If we see in a nut cell, within the cell there is a nucleus, and within the nucleus there is a nucleoplasm, and within the nucleoplasm there is a nucleofibrils. Is the thread-like structure. Nucleofibrils are nothing but chromosomes, and they are present in a spreaded condition inside the chromosome or inside the nucleus. Inside the nucleoplasm they are suspended. They are in a suspended condition, and these are the nucleus. Now, if we will see that what is actually the structure of chromosome, then we will see that this is a chromosome, a single chromosome, and within the chromosome this is the metacentric chromosome, and the condensed, and this is a short chromosome, and this type of chromosome is actually formed during the metaphase stage of a cell division, when cell divides. This is the centromere, and within the chromosome there is a DNA, and where actually gene is present, if we will ask the question, then we will see that within the chromosome there is a DNA, and a particular part of DNA which is responsible for the formation of a particular protein, functional protein, and that particular protein will form a particular trait. Then we will see that that particular part of a DNA is the gene. So in a nutshell, we will see that gene is nothing but a part of a DNA which is responsible for the formation of a functional protein that determine our traits or characters, behavior, whatever may be the. Now, if we will see from the top that where actually gene is present, then we will see that in cell there is a nucleus. Within the nucleus there is a nucleoplasm. Inside the nucleoplasm, suspended conditions, where the nucleus is present in a suspended state. Chromosome is present in a suspended state in the form of chromatin, and that particular chromosome contains the DNA, and a particular part of a DNA is called gene, which is responsible for the formation of a particular protein. Now, if we will see that what is actually the structure of a chromosome, we will see that this is a metaphase chromosome, and this is a chromosome, and few terms we normally confuse. Students normally confuse about few terms of chromosome, as like chromosome, chromatids, chromatin, chromonema, and chromomere. These are the same type of terms and very much confusing. What is actually the chromosome? In case of metaphase chromosome, this is a chromosome, and chromosome is nothing but a structure present inside the nucleoplasm in a condensed condition. During the phase of cell division, they becomes shortened, and the structure is very clear. That is the centromere and two arms. Just before the cell division, they divide longitudinally and form two chromatids. This is the chromatin and this is chromatid. This is actually a single chromosome, but their parts are divided. It divides longitudinally to form two chromatids. This is a chromatin and this chromatid. During the cell division, this chromatid separates and form a single individual nucleus. So, what is chromosome? As we know, as already known, this is a chromatid. Chromosome divides longitudinally to form a two-part with a single centromere. This is a single centromere. And after the cell division, each chromatids will behave as a single chromosome after cell division. Now, this is a chromosome, and if we see that if what is present inside the chromosome, if we see that what is present inside the chromosome, this is a thread-like structure, coiled to form this kind of condensed structure, and this long coil-like structure is called a chromatin, and that particular chromatin is made up of DNA, nothing but a DNA, and DNA wrapped around the protein structure is called a nucleosome. These proteins are histone proteins. These are positively charged. These are positively charged histone proteins, and DNA itself is a negatively charged. So this negatively charged DNA is wrapped around positively charged positively charged histone proteins. So they wrap tightly. However, how much amount of what are the different histone proteins? Histone proteins are 45. Histone proteins are of 4 types: H2A, H2B, H3, H4, and another histone protein is H1. Sorry. And these histone proteins are actually made up of lysine, arginine, and histidine. These kinds of amino acids that are the basic amino acids. For more than 45% amino acids present inside the histone proteins are mainly lysine, arginine, and histidine, are positively charged and basic amino acids. However, the DNA wrapped around this structure, so 4 kinds of histones are there: H2, H2A, H3, H4 as I mentioned, and these 4 kinds of histones present 2 times each, that is form a histone octamer with a H1 lock H1 protein, histone protein. However, so these kinds of structure is chromatin fiber is nothing made up of histone proteins, and these histone proteins together form a structure is called nucleosomes. Here I have mentioned this is called nucleosomes. So this is a chromatin fiber. So I have mentioned chromosome, chromatids, and chromatid. Now 2 terms remain, chromomere and chromonema. In some cases, sometimes these beads-like structure present inside the chromatin is called chromonema, and if we see in an electron microscope, the chromosomes, thread-like structure of the chromosomes sometimes showing dense structure. These dense structure are called chromatin. So beads-like structure are chromomere, and the dense structure are chromonema. So I have already mentioned the 5 terms: chromosome, chromatids, chromatin, chromomere, and chromonema. Now I will discuss about a basic structure of a chromosome. Chromosome is a metaphase chromosome. You will see that the metaphase chromosome is made up of only few parts. 2 end is called telomere. These 2 parts are very vital. In that case, why it is very vital? Because this telomere contains a hairpin loop structure of DNA, so that this part and this part not together, coiled together, or other chromosomes not attached with this chromosome. So this part is very important. It contains the primary constriction region called centromere, and this centromeric region is called primary constriction region, and secondary constriction region is present here. Another region is that NOR, nucleolar organizer region. That is, this part is responsible for the formation of nucleolus, and these are nothing but formation. These are responsible for the formation of rRNA, that is ribosomal RNA. The ribosome, our cellular organelle ribosome is made up of these kinds of RNA, and present these are developed in NOR region. However, if we will see that the centromere, what is actually the centromere? Inside the centromere, inside or in a side of a centromere, there is a special protein called kinetochore. This particular protein is responsible for separation of two chromatids during the cell division. It's called kinetochore. This area is responsible for the attachment of a spindle fiber. During the cell division, from the two end, this is a cell, from this end and this end, this is a kinetochore region, and spindle fiber will attach in that region, and they will separate. They will go in separate. They will just separate. They helps to separate the chromatids into different region of a cell during the cell division. However, if we will see that what is actually the different chromosomal types, we will see that four types of chromosomes are there. There is the metacentric, if centromere present at the center. Sub-metacentric, it is present just behind the, near to the center. Acrocentric, that is the centromere is present in the neck region. And telocentric, if centromere present in a telomere region, it's called telocentric chromosomes. Thank you.